Okay, we're going to make a start now. So welcome everyone to our A Level AQA Biology session. In today's lesson, we are going to be looking at the humoral response. We're going to start off by looking at the response itself. Then we're going to focus a bit more on B cells, and then we're going to finish off with a couple of exam questions. So in our last lesson, we looked at the cell-mediated response, and in the lesson before, we looked at um, phagocytosis, okay? And this is basically the last stage that happens within the process. Okay, so first you have your phagocytosis, then you have your cell-mediated response, which is a response which involves T cells, and then you have your humoral response, which involves your B cells. And if you combine all of that together, your phagocytosis is the part where your pathogen enters the body, and once it entered your body goes and attacks it. Your T cells is the process of it's the intermediate process of producing your antibodies, and then your B cells are the ones in which your antibodies are produced in. Okay, so we're going to start off by again looking at the specification. So far in this area of the specification, we have looked at this part that we looked at in our first lesson. We've looked at antigens, how you define an antigen. Remember, an antigen is just a foreign protein which stimulates an immune response. We've looked at phagocytosis, as you can see there. We have looked at T cells, as you can see in our last lesson. And today, what we're going to look at is we are going to be looking at B cells. Okay, and then in our next lesson, we are going to be looking at antibodies, immunity, vaccines, that sort of stuff. Okay, so then that would finish off this area of the spec. Now, most exam questions that get asked from this area of the spec, they normally come from the vaccines part, um, vaccines and immunity, that sort of aspect. There's actually very few questions which come up in terms of T cells, B cells, phagocytosis. However, they do come up. Okay, so it's really important that you do understand the concepts behind them. Okay, so far what's happened, we looked at this in our first lesson. This is our phagocyte, and the pathogen has entered our body. Now, the pathogen releases certain chemicals, and that will bind to the receptors that are present on our phagocyte. Okay, so as you can see, the receptors are present there on our phagocyte. The pathogen has specific antigens present on its surface, and that's going to bind to the receptors that are present on the phagocyte. And once bound, the phagocyte is going to engulf this pathogen. So it's going to engulf the pathogen. Now, once it's engulfed the pathogen, it is held within a phagosome vesicle. So as you can see, we have a circle around the pathogen, and that's your phagosome vesicle that it's held within. In your phagocyte, you also have lysosomes. And in your lysosomes, you have enzymes, which are hydrolytic enzymes. They are called lysozymes. And what happens is that the lysosome approaches the phagosome vesicle, and it fuses with that phagosome vesicle, and you form a phagolysosome over here. You don't need to know that term. You just need to know that the lysosome fuses with the phagosome vesicle. And as you know, the lysosome has lysozymes in it. It has hydrolytic enzymes within it. Those hydrolytic enzymes, they break down the pathogen. As you can see, the lysozymes break down the pathogen. Okay, and then finally, the waste products are released, and because the pathogen had antigens present on its surface, the antigens are presented on the surface of the phagocyte. As you can see, these are antigens which have come from that specific pathogen. So now the phagocyte has become an antigen presenting cell. It's presenting the antigens from the pathogen. So it's an antigen presenting cell. Okay, so you've got your antigen presenting cell there. Now, once you've got your antigen presenting cell, what happens is that your T cells, they are also circulating the blood. And your T cells, your T helper cells, they will bump into this antigen presenting cell. And they will get activated once they have bound to an antigen presenting cells. Because of these antigen presentation from the phagocytes, they will activate T helper cells. Okay, and then these T helper cells, they will activate B cells, they will produce more T helper cells, they will stimulate more phagocytosis, they will produce cytotoxic T cells. Okay, and those are just some of the functions of T cells. So that's your cell-mediated response, the activation of T helper cells. Yeah, your T helper cells are the ones which are going to produce your cytotoxic T cells. Okay, these are the ones that will kill the pathogen, and they will remove any toxins that they are producing. They will counteract them. They will also go and they will produce more T helper cells. So we have enough bodies forward to fight off the pathogen. And they will also stimulate more phagocytosis. Okay, so that's what happens in your cell-mediated response. Okay, so this image is showing phagocytosis, and then what I've written in red over here is showing your cell-mediated response. Now, after that has happened, why do we need another response to finish it off? Well, once the pathogen has entered our body, we don't want our body to feel ill once again. We want to be immune to the pathogen which has entered our body. And that's why we have B cells. These B cells are the ones that produce our antibodies, and those antibodies, they are then stored in our body as memory cells in case that pathogen reattacks in the near future. Okay, so for example, if you get flu once, you're not going to get the flu again straight away. The reason why is because your body would have already produced your antibodies against the flu, and then if you do get flu immediately after, those antibodies will already be prepared to fight off the pathogens which have entered your body. That's why you have your B cells. Okay, so in terms of B cells, we know that all lymphocytes, they are made in the bone marrow. B cells, they mature there as well. Okay, that's different to T cells. Remember when we talked about T cells in our last lesson? T cells, they are produced by the bone marrow. However, they mature in the thymus. So they mature in the thymus. And the best way to remember this, T for T cells, T for thymus. Now, B cells, they are also produced in the bone marrow and they mature in the bone marrow as well. They are contained within your bone marrow. And a humoral response involves your B cells and antibodies that they produce. Okay, so we have your phagocytosis to start off with, then you have your cell-mediated response, which has your T cells in it, and then you have your finishing response, which is your humoral response, and that includes your B cells. So what we're going to do is we're going to start off by looking at the process which happens after your cell-mediated response. Okay, that's what we're going to start off with. So first of all, pathogen has entered your body. Now, obviously, the pathogen is going to be engulfed by your phagocytes. However, the pathogen is also going to release some antigens. And those antigens, they're just going to be free floating within the blood. Okay, the pathogen, the pathogens are antigens. They're just going to be free floating within the blood, some of them. And when a B cell sort of comes across that pathogen, what happens is that it takes the antigen into itself by endocytosis. This is just a process by which the antigen moves into the B cell. Okay, so it's just a process by which an antigen moves into the B cell. Okay, so process by which antigen moves into B cell. Okay, it's endocytosis. When something is released, that's exocytosis. Exo for exit, and endo for in. Okay, so that's the process by which the antigen moves into the B cell. So at the moment, we just have B cells just floating around our body, and we also have these antigens which have come from that pathogen also floating within the body. When this B cell comes to an antigen, it will take in that antigen by endocytosis. Okay, and what it does is that the B cell will become an antigen presenting cell as well. So this is our B cell, and this is our antigens. Okay, and then we have certain receptors on the B cells as well, just like T cells had. There are certain receptors on B cell, and then this antigen will bind to this certain receptor on the B cell, and then these antigens will move into the B cell by endocytosis. And then what happens is that the B cell will then go on and it will present the antigens on the surface of the B cell. Okay, so these are your antigens. So it's become an antigen presenting cell. Now, our phagocyte was also an antigen presenting cell, and it was activated by a T cell. And that's exactly what happens over here. Now, this B cell, which is an antigen presenting cell now, it will combine with a T helper cell, and that T helper cell will activate this B cell. Okay, so just like the phagocyte became an antigen presenting cell, it was presenting the antigens on its surface. And when it was presenting these antigens on the surface, it was activated by the T helper cells. They bound to the antigen presenting cell, activating it. Over here, the B cell takes in the antigen, presents it on its surface, and then it is activated by a T helper cell as well. Now, once it's been activated, it goes through clonal expansion, and this is basically just mitosis. Okay, so this is just mitosis, and what happens is that loads of copies of these B cells are made. Okay, so lots of copies of B cells are made. Okay, so that's basically what happens. So B cell has been activated by your T helper cell, and now it has been stimulated to undergo clonal expansion, which is going through mitosis to produce a lot of copies of itself. Now, once it's done that, um, it would have produced a large number of cells, and then those cells they will differentiate into either plasma cells or memory cells. Okay, and we're going to go through them in the next couple of slides. Okay, and that's basically your humoral response. It's much shorter than your cell-mediated response, but in your humoral response, you've got your B cells. They become antigen presenting cells by taking in the antigens from the pathogen and presenting them on its surface. The B cell is then activated by a T helper cell, and that stimulates the B cell to undergo mitosis and clonal expansion. Okay, and that produces a lot of B cells, and then that large number of B cells that you have, they will either differentiate into plasma cells or memory cells. Okay, so that's what happens in your humoral response. We're going to go through a diagram now just to visualize it a little bit more. So over here, our B cell has been activated. Okay, so as you can see, it's got the antigen of the pathogen present on its surface. The B cell has been activated. Now, once it's been activated, the T helper cell will come and it will bind to this B cell over here. And once it's bound, well, it's been activated because the T cell has come and bound to this B cell. It's been activated. So this B cell has been activated over here. And what happens is that this B cell multiplies in numbers to produce lots of copies of itself. It goes through clonal expansion. Okay, now we have a lot of copies of our activated B cells. Now these B cells, they will either differentiate into plasma cells or they will differentiate into memory cells. Those are the two different types of cells that they can differentiate into. Okay, and this is all that happens in your humoral response. Okay, so your T helper cell activates your B cell after the B cell presents the antigens on its surface that stimulates the B cell to undergo clonal expansion and produce a large number of copies of itself. And then those B cells will either differentiate into plasma cells or memory cells. Okay, and we're going to be looking at plasma and memory cells on the next slide. Okay, so plasma and memory cells. Now, plasma cells, they are the ones which produce your antibodies. Okay, so remember, antibodies are really important to get rid of that pathogen and to make sure that your immune system has some form of immunity to the pathogen which has just entered. That's what your plasma cells do. Okay, they will produce your antibodies. Now, your memory B cells, they will store the antibodies. They will basically, they will have a store of they will they're basically the memory of the pathogen. Okay, they know how to fight off the pathogen and they're going to be stored safely within your body in case that pathogen re-enters. And if that pathogen does re-enter, these memory cells, they will divide rapidly into plasma cells. And once they've divided rapidly into these plasma cells will be able to produce a large number of antibodies. Okay. Now, the reason why we have two forms, we have plasma cells and B cells, is because memory B cells, they are stored in your long-term memory and they will live in your body for decades. For a really long time, they will be stored in your body. Plasma cells, they are just the antibodies for that sort of initial moment, that moment in time to produce antibodies. That's why your plasma cells are there. They are very short-lived. Okay, so your memory B cells are the ones which are stored in your long-term memory and they can live for decades within your body. And when needed, they will divide into plasma cells, which are the ones which produce your antibodies. Okay, so plasma cells produce your antibodies. Your memory B cells are the ones which divide into plasma cells when reinfected with the same pathogen in the future. And memory B cells, long-term memory, live in your body for decades, whereas your plasma cells are short-lived and it's only for that specific situation. Okay? So going into memory B cells a little bit more, and this is the last bit of sort of information that you need before we move on to exam questions. But memory B cells, they produce plasma cells when they collide with the antigen that they have previously been encountered with. So for example, let's talk about COVID. In our body, when we've had COVID, our bodies would have produced our normal sort of antibodies and memory cells against the COVID virus. Okay, now in the future, if you were infected with COVID again, your sort of memory cells will get activated because they will combine with the antigen from the COVID that has entered your body. And then they will produce your plasma cells, which will produce your antibodies to get rid of that COVID fast before you experience any symptoms. Okay. So this causes a lot of antibodies to be produced so rapidly that the pathogen is destroyed before any symptoms occur. So that's the main purpose of your memory B cells. And because of this, that's why we've been able to develop vaccines, because vaccines contain the dead or inactive form of the pathogen. That means your body will treat it exactly the same as the normal pathogen and it will produce antibodies and memory cells against the pathogen itself without you feeling sort of very ill in the first place. Okay, and because your body has been exposed to this virus, this pathogen, sort of normally, and it's just entered your body, and your body has gone through the work, it's done the work, and it's produced your antibodies and your memory cells through being exposed to this pathogen. This is an example of active immunity because your body has actively made the antibodies, the plasma cells, the memory cells by itself. It's not taken any other needs to produce these memory cells or antibodies. That's why this is active immunity, because your body has actively worked to produce these memory B cells.